everyone, I'm Roya, and me and Jake recently completed a TAS2R38 gene analysis lab in which we extracted and amplified our own DNA using PCR, and then we analyzed our DNA samples under gel electrophoresis to determine our genotype for the PTC tasting trait. And the origin of studying the PTC tasting trait is from the late 1920s, where two scientists elicited different responses to tasting PTC paper. One tasted extreme bitterness while the other tasted nothing at all. From that, these two scientists were foundational in later studies, which concluded that PTC tasting is inherited in a Mendelian inheritance pattern linked to the TAS2R38 gene. So Unit 1 of AP Biology, which is what I'll be connecting our lab to first, is the chemistry of life, which provides an overview in the main macromolecules of life on Earth, one of which is proteins. Proteins are polymers made up of monomers called amino acids, and the structure of a protein is dependent on the pattern in which it folds. And the pattern of folding in a protein is dependent on interactions between a specific chain and order of amino acids. And connecting protein structure to this lab is pretty focused on taste perception, because the perception of taste is dependent on the reception of a chemical signal, and proteins are responsible for receiving a chemical signal. So in this case, gustatory cells or taste cells contains proteins that are specialized for receiving different chemical compounds found in food, in this case PTC paper. The shape of the surface protein that is derived from its amino acid order determines what flavor a certain cell is specialized to receive. In this lab, the ability to taste PTC paper depends on whether or not you have proteins shaped to receive a bitter signal. Non-tasters do not have proteins that are shaped and structured to receive a bitter signal from PTC paper, while tasters do have proteins on the surface of their taste cells that can receive a bitter signal from PTC. And this is all because of the amino acid interactions that allow for a protein to be formed in a shape conducive to binding with a PTC bitter signal, which will later on connect with Unit 4.
Unit 2, Cell Structure and Function. The TAS2R38 gene binds to bitter molecules due to its structure. There are three possible genotypes for the PTC tasters. T, which is when the PTC is very bitter. T, which are where you can taste the bitterness, but it's not overpowering. T is when you cannot taste the PTC. PCR heat shock is used to break the cell membrane down so the DNA can exit for DNA amplification. Heat shock also transforms DNA to be single-stranded. In the lab, we need to amplify the DNA because there is not enough of it in order to determine the genotype.
Unit 3, Cellular Energetics, provides an overview of photosynthesis and cellular respiration. However, enzymes are key in the functionality of both of these reactions, as well as the PCR polymerase chain reaction that occurs in this lab. Enzymes are proteins that catalyze or speed up chemical reactions through the binding of a specific substrate to the specific active site. These active sites are uniquely shaped to fit a specific molecule. So in the case of this lab, polymerase chain reaction in our PCR requires special enzymes called restriction enzymes. Restriction enzymes act with high specificity, recognizing an exact DNA sequence. In this specific lab for the TAS2R38 gene, this restriction enzyme targets the sequence GGC, acts as molecular scissors, making a cut between the nucleotide bases, producing two fragments from the original genomic DNA sequences. The action of this restriction enzyme is dependent on its active site being structurally and chemically complementary to the GGC site. So the reason non-tasters have a phenotype different from that of a taster is because non-tasters do not have the GGCC sequence required for a restriction enzyme to make a cut at that exact sequence, because the restriction enzyme cannot recognize a different mutated sequence and therefore cannot act on the mutated allele that is present in non-tasters. Unit 4 is an overview in cell communication, which is broken up into 3 steps: reception, transduction, and response. In the case of this lab, PTC acts as a ligand that binds to a specific receptor on the surface of a gustatory cell that was encoded by the TAS2R38 gene. This protein activates the receptor, which is a form of a G protein coupled receptor, which in turn activates the an enzyme to catalyze the production of a secondary messenger. The secondary messenger then continues to activate other molecules inside of the cell in the process of amplification, which all occurs during the transduction phase of cell communication. In the case of tasting a bitter molecule, these secondary receptors tend to bind to ion channels, which trigger a movement of ions from the extracellular space to the intracellular space. The increase in intracellular ion levels leads to the fusion of vesicles on the cell membrane and the release of neurotransmitters, which go to stimulate nearby sensory neurons. When the sensory neurons travel to the brain, it triggers the ultimate response, which is the perception of a bitter taste.
Unit 5, Heredity. Children are passed down 23 chromosomes from each parent. The different types of genetic variation are mutation, which is changes in the DNA nucleotides, independent assortment, which is when the chromosomes are randomly sorted into gamete cells. Crossing over, which is when homologous chromosomes exchange genetic info. Random fertilization, which is when a random sperm meets a random egg. Gene flow, which is when allele DNA flows between different populations. And sexual reproduction, which is when two random parents create a genetically unique offspring. The picture to the right is of a gel electrophoresis. This uses the polarity of DNA to our advantage. Those rods send an electrical signal through the gel that pull the DNA down based off how big they are. In our gel, 3 lines is a heterozygous capital T lowercase t. 2 lines is a capital T homozygous dominant. And 1 line is lowercase t homozygous recessive.
Unit 6 covers gene expression and regulation, but also has a lesson dedicated to biotechnology, which is applicable in the context of this lab. In this lab, we use polymerase chain reaction, or PCR, which is a biotechnology technique used to amplify specific DNA sequences, in this case, the TAS2R38 gene. The process of PCR involves repeated cycles of denaturation in which heat separates the DNA strands, annealing, where RNA primers bind to target regions, and extension, where DNA is replicated using DNA polymerase. The cycle repeats multiple times, which ultimately yields millions of copies of the DNA segment. In our lab, we use PCR to amplify our personal sample of the TAS2R38 gene. Our initial sample of DNA would have been too small to be visible under gel electrophoresis, and therefore PCR was necessary to amplify our own fragments until they reached a concentration visible under a gel electrophoresis. After amplification, we used gel electrophoresis to visualize our genotypes. Different banding patterns on the gels revealed whether or not we were homozygous dominant, heterozygous, or homozygous recessive for the PTC tasting trait.
Unit 7, Natural Selection. Traits vary through population. Usually the trait that gives organisms the greatest chance of survival for their environment are the most common. Individual organisms don't evolve independently. Populations of organisms evolve. An example of a trait that humans have that's better suited for their environment is the ability to taste PTC. We have this adaptation to prevent us from consuming poisonous plants. In Northern Europe, there's a lack of poisonous plants, so people from Northern Europe do not have the adaptation to being able to taste PTC. The 5 different types of evolution is natural selection, which is where organisms who have the trait that gives them the greatest advantage will reproduce more. Genetic drift, which is a change in allele frequency. Gene flow, which is the flow of alleles between populations. Mutation, which is a change in the DNA sequence. And sexual selection, where certain traits are more attractive, so mates will reproduce with them more. Unit 8, Ecology. A carrying capacity is the max amount of organisms that can live in an ecosystem based off the resources available. Every ecosystem has to have a carrying capacity. Sun energy starts at the producers or the plants and moves up to the consumers. Biotic factors are factors that are alive, like plants, reptiles, mammals, etc. And abiotic factors are factors that aren't alive, like the sun, the water, temperature, humidity, etc. The water cycle includes evaporation, which is where water transforms to gas, transpiration, where plants give off water in its gas form, condensation, where water vapor turns into clouds, precipitation, where water is in its solid form or snow. And the different ways that organisms interact with each other is predation, which is predator versus prey, competition, which is when two organisms compete for the same resources, and symbiosis, which is where both organisms benefit from the relationship.
This year, the world saw its first successful use of CRISPR gene editing to treat a genetic disorder in an infant. CRISPR uses a modified bacterial defense system to alter DNA at a precise nucleotide sequence, which offers potential cures for inherited diseases. However, the technology raises some serious bioethical concerns. One major risk is off target effects. CRISPR can miss its intended site and alter the wrong DNA sequence, which can potentially introduce harmful mutations. Supporters of the CRISPR technology argue that for patients with severe or fatal conditions, the benefits outweigh the risk, as gene editing may be the only hope for a cure. However, critics warn that CRISPR is still too unpredictable, and errors in editing could lead to long term complications.
Human embryos are cloned to harvest stem cells to save lives. First, scientists inject an egg without DNA with the patient's DNA, and then an embryo forms. It is then grown in a lab until it can produce the stem cells needed. This is important because these cells perfectly match the patient rather than a donor cells which will semi-match a patient. However, this does raise ethical concerns over the fact that life is just being created to be destroyed. From one perspective, we can see that stem cells can be harvested to save so many lives. But on the other hand, we would be creating life and then end it. Life shouldn't be created for a short period of time and then thrown out because we have no more need for it.